Okay, sorry. Okay, good afternoon, everybody. So we'll wait a little bit, see whether we'll have more students coming in before we start. Okay, all of you can hear me, is it okay? Okay, good. Now, this course is the LIFS4950 new chemistry. My name is Kenny Zhong, and from the Division of Life Science. I'm sure a lot of you are from Life Science, but I haven't checked yet. And as you know that we are kind of in the special environment that we have to do the course again. Well, last year the course is face-to-face, but this year every class is so far is online from Zoom. So we will see, but especially for those assessment method, we most likely need to do the online assessments. Okay. Now let's go through the course outline. I upload the course outline at the Canvas already, and also this PowerPoint file. Okay, I would also do the same for all the classes. Upload those, and then you can get that before we start the class. But maybe it's a little bit different now that you cannot, add notes at your computer unless that you have two iPad or computer that you can work on. But still, I will upload the files before. So just some of the learning outcomes, as you know that all of the classes have learning outcomes, and the most likely that this class is focused on the new chemistry. As you know that the neuroscience actually include a lot of different areas, and in this course we mainly focus on the new chemistry. And then today's introduction, I'll tell you what do we mean by that. Okay. So actually, new chemistry is closely related to the function of the brain, and in the courses of this class, and you know that actually it's not just something that really academic. It's actually related to a lot of your thinking and so on. Okay, so those are the learning outcomes. Explain the basic concepts and principles of new chemistry and its relationship to the basic function of the brain. Advance in the new chemistry and how that actually relate to different diseases. So if you know a lot more about the new chemistry, then you can make some changes in your day-to-day decision making and problem solving as well. So actually, at the during the course, I will tell you a lot that how that that can impact the society actually. Okay, that's the last one actually, how the scientific research in the new chemistry can influence the society. Okay, the course actually will teach you about how the complicated brain system or the nervous system actually control your thinking and your movement and your daily life. Okay, so this course actually help you to understand more concept about what is neurochemistry and how the transmission of in the nervous system, and how if you affect those processes can modulate the behaviors, or even, in the case of diseases, how that comes out. Now, for the teaching, I hope that, is more interactive. Is quite different teaching online and or compared to face-to-face, because I cannot see your facial expressions. And some of you turn on your video. That is good, actually. I hope that you encourage you to turn on your video as email from the associate provost. That actually can help for me to see your response. I know that might not be, used to, actually, but I will turn on my video all the time, and that's what I can do, and I hope that you can do that as well. Now, during the class, I welcome you to ask me questions, either by chat, or even you can unmute your microphone and ask me questions, okay? That's a welcome, because the things that is the interactive. If I know that you have any questions, then I will know how much you understand the topics, because I'm not quite sure how your background. Some of you might be even in year 2 or year 3. It depends on your years. So I don't know how many of you, actually maybe later next class I can do a polling and then see what is the distribution of you guys that are on that. Now, something that you might want to know better, the assessment scheme. Okay, we have 2 assignments. The assignment is actually very easy. I will give you a link, then you watch video, and then submit a 1-page summary, and then you'll get it. You get the each one you have the 5% of the marks. Okay, so that's a very easy. You just need to do it and submit on time, and then that's it. So this is the scheme that I try to arrange is that not to make you suffer, but make you enjoy the class. Hopefully, try not to do a lot of hard work on you. So those are the easy assignments. Now we do group project every year. Now this time we still can do it. It depends on the progress. Usually the group project will be after the midterm, before the end of the semester. Okay. Depends on whether the improvement in the pandemic, endemic. I hope that we can do a face-to-face one. But if not, then we can still do the Zoom online project. And the project will be group project, and depends on how many students that will stay in this course, because it's still at the job period. So after that, then I will, maybe discuss with you guys how many students per group, and also the assessment method. And usually the assessment will be both by me and by you on everybody. Okay, but the percentage we will discuss it later. And then we will have a midterm exam and final exam. And you can see that the arrangement, I put the group project actually percentage pretty quite more, 25%. So actually it is same as the midterm. And because I want you to, when you do the project, then you actually help you to actively learn some of the component related to the new chemistry. Okay, so usually what we mean by active learning usually is better for students to actually understand the background or the content. Okay, so any questions so far for the assessment? No? Anyone? Let me see. So it seems to be very quiet. Huh? So you're okay with the assessment method? Okay, so mostly because this is 4,000 courses, and it's usually not just by simple exam. Okay. So let me see. Oh, actually, I have some questions. Okay, it's clear. Okay, good. Oh. So this is a course outline. So this is today is the introductions, for example, to the new chemistry. And then we'll start on some of the, now you can see that actually most of the topics we focus on the transmission using chemical component in the nervous system, okay? Now, so we'll first talk about the basic cellular and anatomical organizations of the nervous system. So this is a bit of anatomy that will remove a lot of names and so on. So okay, I have a question from you or student. Will the exam be open book? I'm still thinking. So it depends on how I make the exam. And it might be open book, but I haven't finally decided. Maybe I can do a polling next week and see how many of you like the open book. Open book is not necessarily better, but it's easier to arrange. Okay, I have some chat that you guys are asking questions. Yeah, you're welcome to ask questions, whether it's everybody to or to me directly only. Now, just let me look at it first. For the group project, the group members randomly assigned it. So that one I can randomly assign, or you can arrange your own groups. From previous experience, that you guys like to arrange your own groups. And maybe I will do a polling again next week. How many people in a group? That depends on how many students actually in the class altogether. I tend to make it smaller groups, 3 to 4. It depends if we have too many people, then maybe we have to have more people in a group. So after the abstract period, then I will let you know. Okay, and then slowly. So one of you asked me when will for the midterm. The midterm will be around in October, but I haven't finalized that. In the next few classes, then I will let you know. So one of you asked what will be the group project about. I will announce later that what is the group project and what is the topics. What are the topics for that? Okay. We have consultation period for the group project. What do you mean by consultation period? Maybe the student can elaborate that a little bit more. What do you mean by consultation period? Will the group members randomly assign, or we could form a group by ourselves? For the past experience, I let students to form their own groups, but if the student cannot form the groups, then I will help the other students to form the groups. Okay. So I will release more about the group project as soon as that we have the adjunct period stop. So we still have the adjunct period until until next week. Oh, the consultation period. It depends, because it's I don't need you to submit a draft. You know, it's that the project actually is more like a presentation. Actually, I forgot to tell you, it's a presentation. Yeah, when we announce the topics and the details of project, then I'll give you more details, and you can all of you can ask me more about the details. Is it okay? Yeah. So because now you don't know the format, you don't know the details, then you have a lot of questions. But once I release those, then you can ask me for more details. Is it okay? All right. So there are some reference books, but most of them you don't quite need them actually. So more is the PowerPoint should be fine, more than enough. But those are just for reference if you want to check some of the details. Okay, actually we cover quite a bit of information in those PowerPoint already. So I try to limit the components that we cover. Okay, so that you have overall ideas about the nervous system and the neurochemistry, because most of you are not may not need those details in the future. But just for the class, that if you can have some ideas about some of the things related to, for example, for drugs, how different drugs that abused by teenagers nowadays, or not just teenagers or adults, actually how do they function? Okay, and then when in the future, for example, if you are in law enforcement, then you know that actually why those drugs are bad for you, or if you're working in other cases, or in the future, or in the far future, when you have kids, then you can also explain to them why they're bad. Okay, some of those actually. Okay, those are, I hope that at the end I can give you some of those approaches. Okay, back to the content outline. So we will first look at the anatomical organizations of the nervous system first, and then we'll talk about the neurotransmitters and receptors in the nervous system. So actually, the neurotransmitter and the receptors are the very complicated system in the nervous system that enhance how the neurons communicate, and then actually make you can function. So if those systems are being hacked, and seriously mean the hacking of those systems by different organisms, that can result into a dysfunctional. For example, all the toxins, they are actually acting on a lot of those toxins that affect nervous system. Actually, they're acting on the receptors or acting on the neurotransmitters. Now, neurotransmitters are the chemicals that used to transmit signal between the neurons. Okay, so we'll talk about those, and in each one, then you will see that I will talk about how they are being produced, they're stored, they're released, and then how they're functioning, binding to the receptors, and so on. Now, there are different classes of neurotransmitters, so that makes the system so complicated. For example, there is acetylcholine. This is one of the most studied neurotransmitters actually that control your muscle contractions. Okay, your muscle contraction is based on this, and if there's any problem with that, then you will become paralyzed. You cannot move anymore. Catecholamine. This is another class. It's not just one class, it's several classes. And actually that controls some of the addictions and also other behavior. For example, whether you're happy or not, that is also controlled by the catecholamine. And then there's amino acids. Amino acids is the one that actually you might learn about it. One of the examples is the glutamate. So glutamate is one of the most common amino acid. Okay, it's a non-essential amino acid. So what does it mean by non-essential? You still remember? Anyone? So you give back the biochemistry 1 to Professor Chia. So we have some interesting. So you can get it from food. You cannot synthesize it in your body. Can obtain from food. Well, seriously, you guys are actually passed back to thesis from Professor Cole. Yes, one of you got right. So the non-essential means that you can produce it by your metabolic pathway. Okay, so most common is from the Krebs cycle. The essential one, yes, you're right. The essential one is the one that you cannot synthesize in the body and only attend from food. Okay, good. Some of you still remember after the exam. Okay, good. So actually, the amino acid is very important because when we study about it, actually, it is the one that actually control your working system. So if this amino acid neurotransmitter is blocked, then actually you cannot function. So you become unconscious. Okay, so that is very important. Gaseous molecules as a neurotransmitter, there will be nitric oxide. Nitric oxide is NO, then you might have heard about it. And actually it is some people will say that it is a neurotransmitter. Some people will say that actually it is a neuromodulator. But but it is also a very important molecules in the neurotransmission. Now, after we talk about all those neurotransmitters and their system, then we will talk about the neurochemical basis of learning, memory, and behavior. So this is actually another topic and how we can get the idea of the neurotransmitter can affect the learning, memory, and behavior. So that's the reason why that actually some of the drugs actually can affect your brain functions. One of the most good example is coffee. Yes. Okay. Coffee is actually one of the most commonly used psychological chemicals that can, make you more focused. Yes, caffeine. Yes. It's very interesting. Okay, but that is not a very strong modulator. There are actually some other drugs that can even make you more focused, that you can study better. But the problem of those drugs that usually they have side effects. That's the problem. Okay. Then after and then we'll talk about neurochemistry of addictions. Now those will be the topics related to the project, and then you will know that actually about those later. Okay. Narcotics, abuse the drugs. How many abuse the drugs? So you know that actually how which system that they're acting on, and then see how you can what are the bad consequences of them. Then we will talk about the pathogenic mechanism of the neurological diseases. Now those diseases actually give you an idea of so excuse me, how that the dysfunction of the nervous system will have the consequence for the nervous system. Okay, so some of the time is the loss of some of the neurons, sometimes it's the imbalance of neurotransmitters and so on, okay? If you have time, we'll talk about some of the novel approaches in the treatment of those disorders as well. So those are the topics that we'll cover, you know. It's overall from the very basic and then to the chemical natures of the neurotransmitters, and also some of the bigger picture of how the memory formations and also some of the neurological diseases, okay? So, because this is the introductions class, so we'll just talk about a little bit about, you know, origin of the nervous system. So even though we still don't know how that actually developed, but we believe that actually developed firstly in some of the simple organisms. For example, some of those, you can see that those are in the sea, jellyfish and so on, until they go to the fish and so on. Now, the nervous system is supposed to be, you know, receiving the outside information and then converted to the cell, and then for appropriate actions to respond to any stimulus, okay? So it's supposed to make the organism better fit to the environment. So some of those examples in the jellyfish, in which that, you know, there's some cell that detect the movement or outside stimuli. For example, if the jellyfish catch a fish, then the muscle will pull the fish to the mouth. Okay, this is the simple. So in which that you have some of the detector, okay, detections, and then that related to the muscle, for example, and then that will give you the response to the system. Now later, that will develop further, in which that there might be interneurons that can relate more information, most likely to integrate the information together and then pass it to the muscle. So just like the reflex that we talk about, then you see that actually it is there's some simple linkage and there are some more complicated one. And that's the reason why they slowly develop and then into a more advanced system, just like us. Now in between, for example, a lot of organisms, they still have, you know, for example, if you have pets, do you have pets? I have a cat, actually. My cat is, sometimes I think that she can understand what I'm talking to her, actually. And she responds, actually. But compared to us, we have a much higher cognitive functions compared to, you know, animals. But animals still, they have a similar arrangement of the nervous system. That's the reason why that, you know, we have a lot of animal models use it to study the neuroscience. Okay, so that's some of the very basic ideas about, you know, how the nervous system is being developed. So brain and behavior. So long time ago, so in the Western and the Eastern culture. So for Chinese culture, if you still remember or you study, well, people know that before, they thought that which part is thinking, doing the thinking. So it's a long time ago when people are not, there's no science yet. Yes, the heart. Yeah, so that's the reason why that, you know, when you lost your boyfriend or girlfriend, then you got a heartbreak, you know, so the heart is feeling painful. So that's the time actually in Asian thinking, actually the heart is doing the thinking. So it's here, it's not here actually. Now, but then science started to develop, and particularly in the Western countries, because this is the development of the science. So you know that actually what is at the very beginning, You know who will make the best biologist? As a biologist, one very important skill for them. Maybe you have, when you start studying biology, actually you might have done the same thing, similar thing. Not physician. Physician is another thing. So it's doctors that, you know, those are different actually. Biologists. Yes, good, observe. So if you observe and then cutting people. That's good. That's true actually, part of it. But after cutting, what do you do? Forensic pathologist. I guess you watch a little bit too much the TV program, huh? That's can be a part of it. Yes, see the internal structure. Yes. Finding the origin of knife. That is a little bit more philosophical. We start with something very simple. So maybe you guys are in the digital age. So after you observe, test the function, well, no, not that far. Well, not that far yet. The very ask questions, that actually is the next one. But so one of your students got observe. Yes, that's good. So after observe, what do you do? Making hypothesis? Not yet. Very good. Come on. Oh, good. Now, good. Wait. That's right. We call the observation drawings. Yes, that's the drawing. And then find the differences, finding patterns. Now, the first thing is observation, and then you record the observations. So at that age, there's no digital photograph. So what you need to do is to draw. So still remember, many years ago when you first study biology, the thing is that you look at the microscope and then you draw. That's it. So that's a drawing. Now at that time, so you see that actually so how do they study the neurons or in the brain? So what they have is get the brain tissue, cut it into very thin slices, and then at that time they just develop so-called the silver stain. So that's reveal the structure of the neurons. So do you heard about silver stain? So do you know the mechanism behind staining? Staining means color. Good. Yes, good. So very good. So the staining actually make certain structure actually. So actually sometimes there's a good part about Zoom. Students seem to more willing to ask questions and give answers. Very good. Selective different colors for the tissue. So now fluorescence is much later. So we talk about some of the very basic one. So because of the chemistry, the chemists developed some dye. well, some chemical, and those chemicals can bind to different molecules. Now, silverstone is the one that binds to the protein, and because of those protein structure, and then you distinguish protein and the lipids. Okay, so this is what happens. So Camillo Golgi, as you heard about Golgi apparatus, so he used a silver stain and then stain the neuron. So this is a neuron from the cerebellum, and this is the neurons in the hippocampus, another structure. So you see that, you know, after staining, the scientists actually can look at it under the microscope, and then they will draw the pictures and then reveal the structure of the neuron. Okay, so now after you have the pictures, then you will ask a lot of different questions, and then you have hypotheses and so on. Okay, so for example, some of the hypotheses, you know, when they look at it, they will see how those neurons actually connect together. So those are the hypotheses or the questions they want to ask. And they have experiments to prove it. Okay, so this is the first step. So after that, later the scientists find actually the living muscle and the nerve cells can produce electricity. Okay, so that's another part. So they find that actually those are the structure, and they find that actually the muscle and the nerve can produce electricity. When we talk about the electricity, then you know that actually this is really the case, because electricity, actually the most simple term is the movement of ions. Okay, electrons or even, you know, ions that produce the current. Okay, now so there's a new anatomist. So in the 18th century, so proposed some of the ideas. Three of the ideas that start, you know, the study of the neuroscience. So first of all behavior originated from the brain. Okay, and the particular cerebral cortex actually control different parts or specific mental functions. So for those mental functions that actually get stronger, then that will be bigger. So basically, neuroanatomists actually talk about, you know, is from the brain. Okay, and there are different 35 organs part, and if that part is bigger, then the function will be bigger. So this is called the phrenology, the study of the conformations of the skull based on the belief that it is integrated for the mental faculties and character. Now, obviously, this is not true, but that's how they think. So this is the part that they draw about, okay, at the surface. And for example, if this part is bigger, then you like eating more, okay? Same other relationship and so on. So just like, you know, palm reading, just like palm reading. But that is actually not true, because if your brain is bigger, it doesn't mean that you are smarter, okay? That's not the case. But we know that actually the brain mass is bigger in some people. That actually can help, but it doesn't have the direct connections. But certainly if there's some condition that reduce the size of the brain, then those people might have mental problem. So that's actually true. Okay. Okay, the brain actually has this distinct functional regions. Okay, the brain is actually not just a single mass, but instead it is a well-organized structure. Okay, so first of all, the central nervous system actually is a bilateral symmetric structure with 7 main parts. When we talk about the anatomy, then we'll talk about different parts. For example, the spinal cord, medulla oblongata, pons, cerebellum, midbrain, diencephalon, and the cerebral hemisphere. Okay, so here you can see that this is the brain, very simple organizations in your head. Everybody has a similar structure, although there different. Certainly, there are differences that make up the differences. So our cognitive abilities occur primarily in the cerebral cortex before anatomical distinct lobe. Okay, the frontal, parietal, temporal, and occipital. Okay, frontal planning future action. Parietal actually have the sensory. Occipital is actually at the back, you know, at the back of the head, that control your vision. Actually, a lot of the information from the eye actually going, the raw information is just like, you know, the information just like a camera at the eye, and send all the information to the back of the brain, the occipital, for processing. Okay, so that's the reason why sometimes when you see some things, actually that's not really the true things, because that has been processed by your brain. Temporal lobe, hearing plus learning memory. Emotional is deep structures. There are deep structures actually called hippocampus and amygdaloid nuclei. Amygdaloid nuclei is very interesting. That's the part actually control your fear. It's not in your gallbladder, as in, you know, what Chinese people think. If you have a bigger gallbladder, then you are fearless. That's not true. The fear is controlled in the amygdala, and we'll talk about it when we go to the structure. And actually, very interesting. Some people actually have damaged the amygdala, and then they will lose the fear, in a sense. And when we talk about that, and then I'll tell you one of the interesting story about, you know, when people, they don't have the amygdala, it's not the fear that is not most important, it's another consequence. Okay, cognitive functions are localized within the cerebral cortex. So actually, what we think, you know, for all our higher cognitive functions actually is here in the brain. So that's the reason why any damage to the brain, that will be very bad. And that's the reason why that actually the brain is well protected by the skull. Okay, skull is pretty hard, but when it is being damaged, that will have a very bad consequence. We'll talk about some of those during the class. Now, the organization of the cerebral hemisphere is actually very well organized. So each hemisphere, so it's divided into the left and right. And the left is controlling the right hand side of your body, and the right is controlling your left hand side. The reason behind is actually the motor and sensory neurons, actually they cause in the middle of the spinal cord, and then cause it to the other side of the body. The reason behind, so we don't know actually. So it will be very interesting to solve this, but why is it doing that? We don't know actually. Now, although they are similar in appearance, but they are not completely symmetrical. So the hemisphere, actually the brain, the left and the right, they are not completely, they are similar, but they are not completely symmetrical. So that's the reason why your left hemisphere needs to talk to the right hemisphere. They need to crosstalk. And there's some cases that actually this crosstalk is damaged. And that turns out to be, you know, some people can have their left eye looking at something and then the right eye looking at something, then, you know, they can look at different things and they don't know each other. Okay. But for you, even you put a hand like this and you still know the left and right. Okay. That's a very interesting part. Now the functions are actually physically localized within the cerebral cortex. Now, in your brain, it's not just, you know, the brain is there, and then all the things just like randomly put it there. And actually, we all organize similarly. Okay, the reason behind is actually in the neurologists, they actually can map the site that control the different part of your body. And most of the people will have a similar organization. Okay, so you'll see that this is called the homunculus way of illustrating the location and amount of the cortical areas, so dedicated to particular functions. Now this homunculus, actually you can see here. So why is it so funny? So you have a very big lips, tongue, hands. Now, though this body illustrates how many neurons are sensing this part of the body. So our tongue and our lips are actually very sensitive. Our hands as well. Now the part that's less sensitive actually is the back. So that makes sense because that, you know, the part that actually needs to have the most sensitivity, the one that your hands and not your lips. So it's basically actually how many receptors per surface area. Okay. Now you can test it if you want. This time I always ask students, you need to ask a very good friend to test it for you. Very good friend, because otherwise that may hurt you. So ask your friend to have 2 needles, and then you can close your eye, and then you can put your 2 needles at the place that you want to test, and then you get it closer and closer. Okay, just pinch it like this, closer and closer. And you close your eyes, then there will be one point that the 2 needles, the feeling of 2 needles become 1, because it's too close now that you know that you cannot distinguish it anymore. Okay, and those hands will be more sensitive because hands actually you're working on it. That's the reason why whenever that you hurt your hands, actually it's very painful, your fingers. You know one of the cut that is the most painful. You know what cut is the most painful cut? Your hands. I don't know whether you have it. Yes, paper cut. That's true. Paper cut is actually very painful. I don't want you to try, but I mean, that if you have experienced that, paper cut is extremely painful because your fingers actually is very sensitive. And your lips is very sensitive as well. Okay, that's a part of the reason, maybe the reason why that kissing is actually a very intimate behavior, because you can feel really strongly. Okay, so those are the functions. So now we talk about the building blocks of the brain. So in the arrangement of the class later, we'll divide the anatomy, and then we'll talk about the cell, the building block of the cell. Now in the nervous system, there are 2 major types of cells. One is the neurons, one is the glia cell. So you can see that, sorry. These are the neurons. Neurons are highly specialized cells. You can see that they have different morphology, and it is because of their different functions. Okay, so there's sensory neuron, motor neuron, multipolar interneuron. So you can see that they have the dendrite and the axon. We'll talk more into it in the class. And we also have the glia cell. Glia cells are the supporting cells for the system. We also talk about those. Now, actually, those two types of cells, they occupy half and half of the brain. So that they support the glia cells, support the neurons for their functions. Okay, and the neurons mainly provide the integration of information and transmission of the signal. Okay. So you can see that this is the neurons, and then you can see that if you see those, this is actually the the nuclei of the different cells, including the glia cell. And the red color is neurons. So you can see that not all the red color contain those nuclei. So those are other nuclei. Actually, part of it is from the glia cell. But you can see that from the neurons, it's a very big nuclei. And there are a lot of spots because the neurons actually are actively producing protein, okay, doing the transcription and translation. Okay. So this is another pictures. And you can see that the neurons are close together, and then those are the axon or dendrite, branching to each other. So this is pictures actually is a fluorescence image of the enzyme that this neuron use to produce the neurotransmitters. Okay, so that's the reason why that you can stain that with the fluorescent dye. Okay. Okay, so any questions so far for the introduction? Now those parts we will talk in details in different lectures. So today is just for an introduction for you guys. And then if we can finish earlier, then maybe you can let you guys leave earlier because this is your first day coming back for classes. Hope you still enjoy the class. Okay, so there's as I mentioned before, there's when you see the pictures, one of the questions that a lot of early scientists want to ask is how the neurons are being connected. So they cannot see the very much details behind the connections because the microscope actually is not that good enough to see the synapse. So there are two hypotheses. One is that the neurons actually connect directly together, just like cable. So it's like a computer cable. Another one is actually, they are not. They are just like a gap in between. Now it turns out that actually most of the connections, there's a gap. And those gaps are called the synapses. Now, when we talk about there are two types of synapses, one is called electrical, one is called chemical. When we talk about those, and then you know. Now, the chemicals that allow the transmission of the signal in between is called the neurotransmitters. So, for example, here is the case. The neurotransmitter is released and then pass through the gap and then bind to the receptors. So you may ask, why do you like to do this? Because it turns out that this is a very powerful way to transmission. When we talk about so this is to everyone. That's good, actually. How could the axons and the dendrites keep in place without physical contact? Now, they have kind of physical contact when we talk about, but they have some of the proteins actually hold them together. Now, how close that hold together, that will control how good the synapse is. Okay, that's very good question. Yes, they hold together with some proteins that help, but they still have a gap. Okay, the gap is actually very, very narrow, and you cannot see it in the light microscope. You can only see it in the electron microscope. So when we talk about the synapse, then you'll see. Now, but there are some problems with this transmission way. Can you think of one? Yes, there's a, so, but the physical contact is not the one that transmit the signal. So the oh, that's a very good one. So how does the neurotransmitter get cleared after transmission? That's also a very critical uh, process. And then if you cannot get it clear, then that will be a problem. Yes, degradation that you take. We'll discuss this when we talk about the neurotransmitters. So there's one of the problems with this type of transmission. Can you think of it? So diffusion is slow, but that's the reason there's a limitation on the transmission. Because of the, so some neurotransmitter may be lost to surroundings. Yes, but that's the reason why the gap is so close together. So one of the problems of this type of transmission is that it is prone to interference. You know, the release, the binding, and so on. Okay, so it's whenever you have connective part, then that will prone to interference. And we will talk more about it during the class later. Okay, is it okay? Very good. A lot of you are actually thinking. I was worried about that, at noon time, then all of you will be sleepy after eating your lunch. So far, still very good. So the neurotransmitter are actually a language between the neurons. I always compare this to the computer. Computer transmission signal actually is only 0 and 1. Okay, but in the nervous system, actually, it's not just like this, because we have more than one type of neurotransmitters. And we also have, for each neurotransmitter, there are more than one receptors. Okay, and then if you integrate all the system together, then you have a very complicated mode of information transmission. Okay. So, for example, neurotransmitters, the acetylcholine, monoamine, amino acids, neuropeptides, and gaseous molecules, and we would talk to each of those. Okay, for example, this is the acetylcholine. You can see that actually it's very simple molecules, and it is used to modulate or control the muscle contractions, as you might learn it from other classes. Now the receptors, you can see that for acetylcholine, there are two major types of receptors already. The so-called the ionotrophic receptors and the metabotrophic receptors. And those are the more like the G protein coupled receptors. Okay, so this is the ionotrophic receptors. So the ions it's like ion channels, and we'll talk about it, how that actually functions. And this is the G protein coupled receptors. And then at once, then you will see that actually, if that involves the G protein coupled receptors, then that can also control by the secondary messenger as well. So that can integrate together the signal transductions. So we'll talk about those. Okay. So are you guys still okay? Now, in the face-to-face, usually I will give you time for break, but for online, then because it's not quite meaningful, because you cannot do much, so I will tend to continuous, but I will finish the class earlier. Okay, is it okay? Now, okay, good. Now, so one of your classmates asked, do most of the neuron use specific neurotransmitter or they use different kind at the same time? Now, this is actually a very good thinking. And actually, there's a scientist long time ago called Dell, and then he also asked these questions. So one of your classmates really think like a scientist, famous scientist actually. So there's a Dell hypothesis. Very good. So I will talk about this during the class because it's not that simple that I can tell you yes or no, but basically more than one in some neurons. Okay. Is it okay? Okay. So let's continue. So as I told you, it's very complex, the receptor system, because there are different locations. Receptor can be in different locations. They are very difficult to study. They are not easy to study, but there are pharmacological studies on that. The type of receptor will be according to molecular biology study or pharmacological study. So when we talk about it, then you'll see that what do you mean by molecular biology study? That's more about the sequence, and the pharmacological study is more about the functions. Okay, so you have two issues about receptor. The receptor has to be physical there, or there's a consequence. There's a sequence, sorry, not consequence. And there, by activating the receptor, there will be consequence. Consequence is the pharmacological. Is it okay? So if there's no consequence, then that receptor, whether it is functional, then that's the question. Okay. So there are also autoreceptors and postsynaptic receptors. So the one that release the neurotransmitter, they will have autoreceptors. Means that the neurotransmitter will bind to the same place that is released from. Okay, so if you still look at the pictures here, so you can see that this released to bind to the postsynaptic terminal, but it can still bind to the cell at the presynaptic terminal. So that serves as a regulation over here. Okay. And then there are further classification of it. For example, this is a cholinergic, so using acetylcholine as the agonist. So we'll talk more about the agonist and antagonist as well. So they are divided into two parts and then further divided into different parts. So you can see that there are so many different kinds of receptors. Okay. So that actually make it more complicated in the functions. So we'll talk more details about this later. Okay. So learning and memory from anatomical structures to synapses. So that is actually another big questions about the learning and memory and the synapse. So how can we connect the synaptic structures and the neurotransmitters to the anatomical structures to the higher cognitive functions, which is the learning and the memory, okay? For example, now actually your brain, suppose if you are listening to me, you're actively, working your synapses and so on, So how can we integrate to those? Do we know the detailed mechanism of the memory formations? No, we don't actually. There's still a lot to learn from it. So otherwise, we can have drugs that you can make you better learning. You just take one drug, and then you don't need to take a whole course. You just flip through the PowerPoint, then you remember and understand. So that is not something that can do it. Even though a lot of people want that to happen, but that's not the case at the moment. So you see that actually there are areas in different parts. So I'm not going to talk about in detail, but different parts that control the feeling, perceptions, speech, learning, memory, and skill and movements. Now, something that, as I mentioned before, something that you can remember very strongly, something that you actually don't even remember at all. Why is the case? So for example, I always give student one of the example. If you eat something good, think good, the restaurant, you will remember it very strongly. Well, if you eat something very bad, you still will remember very strongly. Okay, both the case. So because that good food, you want more. Bad food, you don't want any more. Okay, that's always the case. Now, in recent studies, actually, to test whether you believe the memory is actually sitting in your brain. Actually, there are so-called the functional MRI, the functional magnetic resonance imaging, tested in volunteer individuals. Actually, when asked them to think about something, for example, a hammer, then one special part of the brain will light up, okay? And it's always there. So that means that physically, the memory is actually sitting at in your brain. And another evidence for that is actually when we look at some of the diseases, for example, in the Alzheimer's disease, when the brain is actually damaged, those patients will start to lose their memory, okay? So we'll talk about in Alzheimer's in details, and then you'll know that actually that is really the case, in which the brain actually stores the memory of you, your experience, starting from the very beginning up to now, okay? Now, in the molecular level, we know that actually it's stored in the synapse. We have evidence showing that, and we'll talk about it, so-called the long-term potentiations and the long-term depressions, okay? So the neurochemistry of addictions. Addiction is actually something critical for us to survive. That including motivations. Actually, for example, eating and drinking. If you eat something good or you drink something good, you want it more. So that's the same circuit that actually related to addictions. Same thing for long-term survival. For example, sexual behavior, or caring for the offspring. Those are actually a way to make us pass through the our species, What about mental illness? Oh, we will talk about the mental illness as well. Okay, that's an addiction. So addiction is actually compulsive drug usage. So actually that is controlled by the a circuitry, okay? And that actually helps us to survive. We'll talk about it in some of the project cases, okay? So now there are two types of addictions actually, drug addictions and behavior addiction as well, okay? It's not just drug, but behavior as well. But both of it are related to the circuitry, okay? This circuitry. Pain. So we don't have a lot. I don't think that we have time to talk about it, but it's just to give you the example of it. The pain is actually a very important part. So for example, aching, stinging, and also soreness. And they actually mediated by different type of receptors, nociceptors, in which that actually at the peripheral flow actually detect the pain. Okay. So the last slide today, the neurological diseases, including seizures and epilepsy, mood disorders, or mental disorders, attention deficit hyperactive disorder, neurodegenerative disorders. Now, we'll talk about some of those disorders and how they are related to the imbalance of the neurochemistry or the neurotransmitters. Okay? Okay. So we'll stop here. Any questions? Any more questions for anything today? So the video, when this is done from the Zoom, I will post it. The schedule of the assignments, and I will give you the schedule of assignments early. I will post it sometime, okay? But don't worry, I will post it way ahead of the submission day. Okay. Any more questions? Someone asked a very interesting question. Is it possible to have a helmet which could send electrical impulse to our brain to affect our decisions? Well, technology-wise, that's not possible. But what I can tell you is that you can have a helmet to detect the brain wave, for recording. For Neuralink, that's actually active. People are actively trying to do that. So what they are trying to do is to have, this is serious, they are thinking about it, to have a chip pinpoint to the brain and then use that to control, or the helmet to control the robotic arm. So that's for, for those disabled people to do it. Optogenetics will not do it to human. You can do it to mice, actually, or monkey. That's, people are doing that. Now, can that wave give us the detailed information of the activity inside the brain? Now, if just think of it, if they put a helmet at your head and then they can tell what you are thinking, then that will be very good for those people who want to steal information from you. So it's not possible yet. People want to do that, but not possible yet. Can neuron be rewired? When you are young, yes, but when you get old, it's very difficult to rewire it. So that's the reason why that, like you now, it's difficult for you to pick up new skills. Not new skills. I mean, for example, if I ask you to play piano, learn piano, then that would be more difficult. So when you are, mature, then you don't want to rewire it because you have stored the information. Just like, if you learn, once that you learn how to ride bicycle, it's very hard for you to get it, well, to de-learn it, okay? You learn it and then that's it, it's inside. Tensile neurons being stinged. Oh, the sting neurons, those are using antibody. So the neurons actually, because of the specific metabolic pathway, and you can use certain antibody to label those neurons. So when we talk about those, then you see. Well, you already had the course of the Alzheimer's. You'll learn it when we get to that part, if you stay in the course. Fluorescent protein. That one is not fluorescent protein. You can put fluorescent protein in the neurons, not in human. Wow, a lot of questions, very good. Any more? So is there only one type of memory, and what is the subconsciousness? Subconsciousness, I don't know, but there are different types of memory. When we talk about memory, then we'll give you a very brief ideas about it. Memories are actually very complicated, but we try to classify that into different categories. Okay, any more? So I will end it here because you may still have the next class. Okay, I'll see you on Wednesday, same time. Okay, goodbye. Bye-bye.